Good afternoon, ladies and gentlemen. Welcome to this exciting session on what is the future of transplantation. This is the first part of two sessions. The next one will be in October. And today I'm going to be the moderator. I'm a transplant surgeon at Columbia University and a former director of the transplant program. And my co-moderator is Dr. Karin Hehenberger, who is a physician, endocrinologist. She's an MD, PhD. What is most striking, besides being the CEO of LifeBulb, is that she is the recipient of a kidney and a pancreas. So she has a very close touch to all this transplantation matter. We're very gratified to have distinguished panel of physicians and surgeons, really, who have made major progress in the various fields and about which they will comment. So I will start with Dr. Griesemer, who is professor of surgery and transplantation at NYU, an adjunct professor at Columbia, and he's also the head of pediatric renal and liver transplantation at NYU. He's an expert on xenotransplantation, and that will be the focus of his discussion. He will not today speak about tolerance because it will be just too much, so we will probably delay that till next month. On the lower right, and raise your hand, is Dr. Piotr Witkowski. Dr. Witkowski is the renal transplant and pancreas transplant surgeon at University of Chicago. And he's also the head of Pancreatic Islet program and of the Pancreatic Islet experimental program at University of Chicago. And just below me is Dr. Joshua Weiner, who is assistant professor of surgery at Columbia. And he heads the intestinal transplant program. And for obvious reasons, he's an expert on microbiosis, which is related very closely, obviously, to the intestine. He will discuss the whole issue of the influence of microbiosis on transplantation, rejection, and other matters that are concerned with this. We will start the discussion. Now, I'm sorry. This whole program is partially supported by Veloxis, so I should mention that, and I would like to turn this over now to Dr. Hehenberger for a few comments.
I think you said it all. Thank you, Mark, and welcome to all our excellent speakers. We are really excited. These three topics are really top of mind for our transplant community, and as always, we have a number of questions that have been forwarded, and we have presented them to our speakers, so they will take them into account as they present. This session is a little different than our normal sessions. We are going to have you use slides, and there will be time for questions afterwards. But it's also, of course, recorded, so you can see the archived version later on. But thank you, everyone, and please, let's get started. Adam?
I should mention to you also that Dr. Griesemer will have to leave a little early because he has a vital appointment with an airplane.
Thank you, everyone. I do want to share just a few slides to set the table for questions and discussions about some recent developments in xenotransplantation. As most people that have any relationship with transplant know, one of the biggest problems with the field is the shortage of suitable organ donors and donor organs for the number of patients that could benefit from transplantation. And this slide just shows the big gap between the number of people on the waiting list and the number of organ donors that are available. So one of the technologies that the field has been looking into for a number of years is xenotransplantation, which is the transplantation of animal organs into humans. And for various reasons, the field has settled on pigs as the most appropriate donor animal for these transplant experiments and future clinical translation. So there have been recent developments in transplant, spearheaded by a group at NYU, where we have transitioned from testing transplants from pigs into primates to pigs into humans as the final step before performing clinical trials of this technology. And what they have done is taken patients who are brain dead, but are not suitable to donate their organs to other human recipients, and use them to test the initial immune response and survival of pig kidneys from genetically modified pigs into humans. And these experiments were done by taking the pig kidneys out and transplanting them into the femoral vessels, with the femoral artery providing the blood flow and the femoral vein the drainage. And then we were able to cannulate the ureter from the pigs and isolate the urine output directly from the pig xenograft. And that allowed us to judge the function of the pig kidney without any interference. And these are the two initial transplants that were performed at NYU. You may have read about them in the newspapers. They did get quite a bit of media attention. This is the first pig kidney, and this is the kidney 54 hours after reperfusion, showing normal color and no signs of external rejection. The histology of the two kidneys showed normal appearing glomeruli with no evidence of any inflammation or no lymphocytes or white blood cells infiltrating the kidneys, suggesting that at this time point, at least, we did not see any evidence of immune rejection of the pig xenografts. These experiments were also translated to, sorry about the announcement in the background, translated to heart transplants, where two recipients recently underwent heart transplantation from genetically modified pigs. These pigs had 10 genetic modifications prior to the transplant, whereas the pig kidney donors only had one genetic modification, but we removed one pig gene. These pigs had 4 pig genes removed and 6 human genes added to help prevent clotting of the blood vessels and the microvasculature prior to the transplant. And that is the last slide. So that's just to stimulate some discussion and questions. The field of xenotransplantation has undergone, there's a long history to it. The very, very first transplants to humans were actually from animals because of the ethics concerned around using organs from a human donor. And at the time, we hadn't established the criteria for brain death. So in the early turn of the century, there were actually animal kidneys sewn into the vessels of the arm in an attempt to provide renal replacement. Once we mastered the techniques and the ethics around human-to-human transplants, the results were so much better that we sort of abandoned xenotransplant for a long number of years. But the gap in the available organ donors versus recipients has reignited a lot of interest in xenotransplantation. It has been stuck in the preclinical stage where we were doing transplants from pigs to other animals as a model to humans, but now we're on the cusp of pig to human transplants. The most likely organs that will be first, it seems like heart transplant will probably lead the way, given the comments of the FDA and their concerns regarding kidney transplants being able to regulate human blood pressure, human sodium, human volume status, and also metabolizing medications, because many of the medications that we use are renally cleared, at least in part. So there are a lot of concerns the FDA has about how the pig kidney is going to function in the human environment, where they seem to regard the heart more as a muscle that pumps and doesn't do as many hormonal regulations of the human environment. One other possibility for early xenotransplant, not experiments, but transplants, is islet transplant. And Dr. Witkowski may comment on that later, as he is the expert in this panel on islet transplantation. But pig islets do seem to regulate human glucose within an acceptable range of normal values. So it is possible that we will do islet transplants as one of the first xenografts. And one of the advantages of islet transplantation is it can be done through minimally invasive catheter-directed infusions of the islets. And so if the islets fail, there is less of a impact on the recipient of the islet transplants. Whereas if you have a heart transplant and it fails, the recipient passes away. If you have a kidney transplant and it fails, it may require a second operation to actually remove the kidney to prevent further complications. So those are the three organs that seem to be leading the field in terms of candidacy of what are we going to be doing in humans first. Liver transplant seems to be one of the, in my opinion, is one of the last ones that we'll probably address. And the reason for that is there are 500 enzymatic activities that the liver performs. And if there are mismatches between the enzymes of the pig and the human, we could have a lot of problems. In fact, we transplant human recipients if they have one enzymatic defect in the liver. We'll perform a transplant to replace that. Those are typically done in children. So if there are lots of enzymatic defects in the pig liver relative to the human environment, we may be transplanting essentially metabolic diseases to the recipient. So we haven't even gotten to the level of understanding which genes are appropriate and which genes are mismatched for the pig liver relative to the human. So that's going to have to be done before we can even think about clinical translation for liver transplants. So
what question? A question from the audience here. A question from the audience.
Perfect.
The gene editing.
Yes.
When and how does that happen and I have a follow-up to that, is that there are some companies doing work in this space, and you're doing work in this space. And how does that function when it comes to the FDA? Is the company ahead of you, or are you ahead of the company?
So the companies are partnering with us, and in full disclosure, some of my research is supported by one of these companies, United Therapeutics, which has generated the pigs that we transplanted into the decedent, the brain dead recipients, and also the pig where the heart was transplanted into a living person in Maryland. So that company has sponsored a lot of this research, and it just, full disclosure, can be perceived as a conflict for some of the things that I'm talking about. But there are other companies that are also in this space that are editing the pig. And so they're using CRISPR-Cas to edit the germline of the pigs. So these pigs will have these human genes inserted permanently, and if they have offspring, they will perpetuate those genes. And when we remove the genes from these pigs that are deleterious, that's a germline mutation. So they are born with these different genetic constructs. They still look like regular pigs. It's just the cell surface expresses some human proteins that can help control clotting, can help control the immune response against the pig organs.
Adam, can you comment briefly on why those pig kidneys were hooked up to dead people, and why only for 50 days or 50 hours rather than 50 days? Can you do it for 50 days? And the other question that I have for you is how does FDA look upon this whole experimental business, not only from the point of view of the function of the organ, but also from the point of view of possible transmission of viruses or anything particular to the pig? Although I must point out to the audience that the pig is probably the cleanest animal in the animal world, despite the fact that we always think of a pig rolling around in the mud.
Excellent
and the closest to the human, right? In many ways, very close.
Oh, they're cleaner than the human. And also they can be raised without any pathogens.
Right.
Yes. So transplants were performed into brain dead bodies, essentially, because they were legally declared dead, because the FDA doesn't regulate that. Since they're already dead, they don't weigh in on any of the treatments of the brain dead person. So this allows us to get data about how the kidneys or the pig organs function in humans to justify a clinical trial to the FDA. They were done for 54 hours initially because our ethical panel had limited us to that, because that is a typical time in which it takes to, once a patient is declared brain dead, to allocate deceased donor organs. So that's a very standard time that we already keep cadavers on the ventilator, their heart beating and things like that to allow organ transplantation. We have gone to our ethical committee and requested the ability to perform these transplants for longer durations of time so that we can evaluate the effect of the adaptive immune response to xenografts rather than just the preformed innate immune response, which was all we could really achieve with that 54 hours. And quite frankly, the responses that we saw were not surprising and were totally expected. But it was reassuring that we didn't see anything surprising in that regard.
But you expect that if xenografts, not if, when they get to market, I really hope when, there is going to be need for immune suppression.
Absolutely. The immune response against the pig is even stronger than the immune response against a human organ. There's the idea of the perfect match in kidney transplantation, where the genetics are from the donor and the recipient are extremely close, and you don't need to use as much immunosuppression. This is almost a perfect mismatch, where
Right.
things are so far apart we have to use more. And so we do use some strategies to try to decrease the immune response. And these kidneys, they had co-transplantation of the pig thymus, which controls T cell development, and that is designed to decrease the T cell or the white blood cell immune response to the pig's organ following transplant. Now, Dr. Hardy, you had one more question about the pig viruses, and that's a very pertinent thing and something the FDA is very concerned about, because pigs do contain a retrovirus that is in the same family as HIV. Pigs have been specifically bred to be lacking that virus, and you can actually genetically edit that virus out of the pig genome. And that virus has never been shown to be transmissible to humans or primates, quite frankly. But there are concerns about other viruses that we may not know about that could be transmitted from the xenograft donor. Now, using a pig gives us a little bit of reassurance because we have been coexisting with pigs for thousands of years. We have bred pigs, we have farmers that take care of pigs, we slaughter pigs, and we think that if there were a virus that could have been transmitted to humans, it has already happened because we have such close contact. We're not as worried about an emerging viral pathogen like COVID that is thought to arise from an area where humans haven't been. And since we've never been exposed, we had no immunity against those pathogens. Now, the FDA wants us to prove the negative, to prove that there isn't a virus that could be transmitted, and that's really not possible. But we are doing viral surveillance of everyone who's come in contact with the cadaver that was hooked up to the pig kidney to ensure that no virus transmission has happened.
But can't you also use the evidence for many, many years? And I don't know whether it has continued, but pig skin has been used to cover burns for 2-3 weeks at a time. And as far as I know, none of the patients ever got any unusual virus.
That is correct. But the FDA keeps asking, how do you know it can never happen? And the answer to that question can only be, we don't know.
Yeah,
Right.
proving a negative is very hard. I think we need to move to our next speaker. This was a great, and this is a topic in itself that could go on for the whole session. But thank you so much, Dr. Griesemer.
Thank you, and have a safe trip, Adam. Stay around
Thank you,
because
everyone.
now Piotr Witkowski is going to talk to us about islet transplants, and also perhaps we'll mention the possibility of islet xenotransplants, because one of the major issues in transplanting pancreatic islets to correct diabetes is the lack of enough islets supply.
Right. Thank you very much for invitational opportunity. So I will start with explanation what's and why we do islets transplant. So we know we can do the whole pancreas transplant, which is on the picture on the left. When we transplant the whole pancreas with the piece of the bowel from the donor, it requires big surgery with vascular anastomosis, the bowel anastomosis. The success rate still is at 90%. So there is risk of 10% risk of losing the pancreas graft, and there is risk of bleeding and other complications. So it's a major surgery and requires immunosuppression. So in order to avoid the risk related to the surgery, we developed the minimal invasive islet transplantation. So what do? We take the pancreas from deceased donors exactly in the same way if we would take it for whole organ transplant. And then instead of transplanting the whole organ, we isolate the islets in our laboratory. And then we put the islets in the solution and we infuse them through the small catheter, which is introduced by interventional radiologists through the surface of the liver into the vein, which goes to the the liver. So the islets are infused in the liver. They're big enough to stay in the liver. They don't go through the circulation of the liver. So islets grow into the liver, and this is when they function, and this is where they produce insulin and regulate blood glucose. So it's minimal invasive infusion of the islets into the liver. So yeah, we've been doing this for the last 20 years. We've been optimizing this procedure. So what we learned that the islets allotransplantation, islets isolated from cadaveric donors, can provide long-term insulin independence. We know that it's safe and effective for the last 20 years of experience. The disadvantage is still that it requires immunosuppression, as most of us know. It's also provides the risk of the side effects. And as we mentioned, there is a limited islet supply. There is a limited number of deceased donors. But also what's very disappointing is current regulation of the islets. Since FDA has been regulated human islets as a drug, we cannot move to we cannot apply islets as a standard of care procedure in the United States, since we academic institution, we don't have a license for drug manufacturing. So it's a big regulatory problem which hasn't been resolved. And after successful clinical trials, we cannot transition to the standard of care. And this needs to be, and hopefully will be, addressed by the regulators. At the same time, as we mentioned, we're trying to develop a way to transplant the islets in different way with unlimited supply. So there was a lot of work to convert to convert the stem cells into the islets, and eventually two companies, they overcome the challenges, and they conducted clinical trials. So first was the VIA side. So they engineer human islets from embryonic stem cells, and they organized clinical trial. However, it was with limited success. There was some signal of the islets surviving, but they, as far as I know, they didn't provide meaningful function. Now, more recently, Vertex, another company, they developed own line of the islets from the stem cells, and they just started phase 1, phase 2 multi-center trial. And the 2 patients, they announced results in the first 2 patients. One patient has been found off insulin completely after only half dose of those cells. So the results of these trials are very promising. If they're successful, it would mean that the Vertex, the company, can provide unlimited amount and number of islets for the patients, and this can be the procedure can be scheduled, and patients may get it on a regular basis. What's the limitation? So it's unlimited supply. However, at this point, the islets transplantation has been done exactly the same way as we do for cadaveric islets. So islets were infused in the liver, and they require the same immunosuppression which we use for cadaveric islets. So at this point, we only the source has been addressed, but the immunosuppression is still a downside. However, at the same time, this company and other company, they actively working how we can to develop a way to transplant the islets without need for immunosuppression. For example, some pouch for the islets which will be immunoprotective, allowing islets to survive and graft at the same time. So this is something the companies has been working on, but it hasn't been tested yet. And the third step in the next step for the islets, it would be the option to transplant the islets from the pig, which Adam mentioned. So having this company and other companies manufacturing the organs or islets which are genetically modified would be the other option as for islets as a source. However, the challenge of immunosuppression remains to be resolved, and it needs to be tested. So we are excited. I'm very excited to see the first clinical trials testing pig islets, which hopefully will happen soon, but that hasn't been so far. So there is another group of patients with type 1 diabetes who already receive a transplant, for example, kidney transplant. So those patients, they already on immunosuppression. So we can offer islets to those patients without adding much risk. So we know there is around 500 patients who receive a kidney transplant without the pancreas, and they will be potential candidates for islet transplantation. But again, if we have financial means to do it, so if insurance will reimburse it, but the insurance will reimburse it if and when the regulations will be changed and islets will be either approved as a drug, or what we're proposing that the islets should be regulated as any other organ, and this will resolve the problem. So this is an example of the patient who received kidney transplant. He wasn't candidate for whole pancreas transplant because of peripheral vascular disease. And as you see, he lost both of his extremities after the kidney transplant due to poor glucose control. Finally, we were able to secure some funding and we transplanted the islets, and he's off insulin for the last 6 months with optimal glucose control. So we wish we can help more patients if there is a reimbursement system for that. So we currently offer we're conducting 3 clinical trials at the University of Chicago, trying to address the major problems with islet transplantation. So we have a trial when we test Cernova pouch, which so we are transplanting the islets into the special pouch, which is implanted in the abdominal wall, hoping for better, more efficient islets engraftment. In another trial we are just starting, we will be testing new anti-rejection medication to replace Prograf, tacrolimus, which is a strong medication and good immunoprotective, but have serious side effects. So we hope with this new medication, new combination of medication, the immune therapy, immunoprotection, immunosuppression therapy will be less toxic. And we also part of that Vertex trial when we testing the islets manufactured from the stem cells. More details can be found on our website. And this is just to show our patients. We organized islets transplant patient reunion in 2015. Out of those 14 patients, and this time point 13 was completely off insulin. It was 2015, so I want to show you what happened afterwards. So those 2 individuals, they still off insulin. So it's almost 9 years after the transplant. They have transplant 2013. The reunion was 2015. Anyway, so they've been 9 years off insulin after only 1 transplant. Those 2 patients, they will be off insulin 8 years. And actually, this patient just delivered a baby. So we modified the immunosuppression, and she successfully delivered a baby. The islet graft, and baby is fine, and the islet graft is as well. She's still off insulin. So there is another patient 5 years off insulin, and now some of the patients, once they achieve insulin independence, the quality of life, the independence in their life was so improved that they found they set up the families, they got married. So the life quality of life was substantially improved. Now, some patients, they were off insulin for a few years, but then they required some insulin and they came to us for more islets. But again, because it's not reimbursed, we didn't have more research funding, we couldn't offer them islets. So we offered them whole pancreas transplant, and they received pancreas transplant, and they have been off insulin for several years. And
thank you very much, Piotr.
Thank you.
And the last thing only. So we as a transplant community has been informing the regulators there is real need for adjusting the regulations so the islet transplantation can be reimbursed in the United States. So far, we hasn't been successful. Thank you.
So this is an appeal to patients to notify their congressmen and their representatives to get after the FDA to approve this, because actually I happen to know that Piotr has written a lot about it, has testified to Congress about it, and still we are not getting where we should be. Now, just going back to the science a little bit, so could you comment briefly, where do these companies get their stem cells, and why don't they take the stem cell from the patient who needs the islets. That way they wouldn't reject.
Right. So I understand these are embryonic stem cells, right?
They're embryonic stem cells.
So they're all embryonic stem cells.
They all embryonic stem cells. So I know there are other companies working on the stem cells isolated from the patients, but I'm not aware about clinical trials at this point.
Both SEMA and ViaCyte use embryonic stem cells.
Yes.
Yeah. So a question, it is really a shame that the islets are not approved in a way that pancreas or kidney and liver are just used and that they're considered a drug. But if you look at the comparison of insulin independence between a pancreas transplant and an islet transplant, you know, what's the difference there on average?
Right. So the most difficult answer is average, right? So islets isolation and transplantation is a very complex procedure, and it requires a lot of experience to have it done efficiently. So we've been successful, and we have over 50 and 60% insulin independence after 5 years, which is comparable with the whole pancreas transplantation. So the results can be comparable, but of course it requires a lot of experience or work with individual patients, making sure they maintain the immunosuppression in the long term. So the results, long-term results may be comparable in the right settings, but the advantages of minimal invasive procedure. And as far as regulation, I didn't mention that only United States is the only country in the world when the islets are regulated as a drug. In Canada, Europe, Australia, Japan, islets are regulated as an organ, any other organ on tissue for transplant, and it's available to patients. So this is
now one of the things that you haven't elaborated on is also in an answer to Dr. Hehenberger's question, is that the islets actually an introduction to the liver end up being very into a very unfavorable environment that sort of knocks out probably about 50% of what is introduced.
exactly
It's not just a matter of isolation, which there are many units like yours that are very expert at it, but also the problem with destruction of the inoculum. And I would be very curious to know what your plans are for that pouch and the other place, to put it in another place, the islets, so they don't get killed by the inflammatory reaction in the liver.
Yeah, Dr. Hardy, you're exactly right. So we believe, based on the animal studies, that we lose half of the islets after infusion in the liver, and we're trying to find a better place with improved engraftment, and that's why we've been testing the Cernova pouch. It's a prototype. We're still optimizing. We're still not at the same level as liver, but we're working on this, and there are other companies working on that, so to improve engraftment. But the liver has advantage that that islets can be infused in the liver without affecting much of the function. So this is major, and providing instant blood supply. In my mind, providing sufficient instant blood supply, it's a key for islet survival. Therefore, all these technologies we're trying to immuno-isolate the islets for immune protection, they also limit the blood supply and limit the survival. And that's why the ViaCyte trial failed, because they tried to immuno-isolate at the same time. The blood supply was not sufficient and islet didn't survive.
But
They used a macro
can't you
encapsulation
modify-
method versus a micro encapsulation, right? ViaCyte?
Right,
Because there's
right.
yeah.
Right. It's a macro encapsulation.
Yeah.
Either way, any additional separation, isolation,
Yeah.
it's limiting the blood supply, which is a key. So of course, hopefully there might be developed and will be developed a method which can combine the immuno-isolation without compromising the blood supply. And this is the ultimate goal.
Well, one of the things that you have been involved in the past, I know, and I hope that you are now, is to get one of these pouches and get them, put stuff in there that will make blood vessels grow first, put the pouch in before you put the islets in it, let it get vascularized, and maybe even put stuff in the wall that will prevent rejection. But theoretically, it is possible to make something like this. Has that been done?
No, so the concept which you're talking about, this is the concept which Cernova applied. So we implant the pouch ahead of time, allowing for pre-vascularization of the space, and then we transplant the islets. And of course, once we optimize the device and the space, we can add additional factors which can enhance the vascularization and islet survival. And this is the ultimate goal, to have a pouch which can be loaded with the other chemicals or other cells or stem cells or Tregs to immunoprotect and islets, limiting the need for immunosuppression, but also improving the survival. So the goal is exactly what you mentioned, to combine technologies to improve islet survival and protection.
So the obvious end question for you is, when do you think I can get islet? If I have diabetes, when can I get islets instead of the pancreas and end up with similar results?
So you can come to the University of Chicago. We have trials. We're inviting you, and this is we are the only. I mean, there are other UCSF and City of Hope. They have very limited trials, and nobody else has currently the funding to continue the work which we've been doing. So it's.
Is Bernard Herring not doing it anymore?
Who?
Bernard Herring is not doing it.
No other center.
And Ricordi is not doing it.
No, they closed. So it's unfortunate. People who had huge experience and they help patients, they all had to close the lab.
This is a shame. This is really a shame on the US. Right?
It's 20 years of work is wasted, and the field is going down, and we don't see the.
Yeah.
Well,
it's a shame. And I want to just, before we move on to Dr. Weiner, it is such a freedom. It's such a freedom to get a pancreas transplant or an islet transplant for a person with type 1 diabetes. And I think that there's a certain disservice that we are only, dealing with the very sick, as you said, the brittle and the very sick people with type 1 diabetes, because the results are going to be worse, because there's so many other issues that these individuals face. I think that if you were to look at a person with type 1 diabetes who had average A1C, the burden of that disease is so high that they would love to get islet transplant or pancreas transplant if there were no immune suppression and the invasive process would be limited. So, we have some work to do, but it seems like it's at least coming in the future. And I think we need to mobilize the forces here to get it not to be a drug.
Yeah.
Thank you.
That I think is key, because I happen to have been involved with Piotr when he was appealing to the FDA. Yep. And it really, from my point of view, it was a painful experience. So I can imagine how painful it was for him, who was really the leading person to do it for the United States. So thank you for the effort. And let's now turn to the new subject, relatively new for transplantation. And that's the microbiotics, and Dr. Weiner is one of the experts in this field. And I would really like to hear the introduction and the reasoning behind it.
All right, well, thank you, Dr. Hardy. Thank you, Karin. Can everybody hear me, by the way?
Yes.
Yes.
I don't have slides to avoid filling your screen with pictures of poop. And also, it gets very technical very quickly. So I'm going to talk about concepts. I'll start with intestinal transplant, where this is the most relevant, but unless you are one of the 100 people in the country who received an intestinal transplant in the last year, I realize that may not be relevant to you. So I'm going to broaden this to other types of transplant where it still remains relevant. In the setting of intestinal transplant, there's a lot of talk about the role of the microbiome and rejection and other immunologic events after transplant. For obvious reasons, the intestine is full of bacteria, some that live there naturally. A lot of those are called the commensals, and some that, as you can imagine, are pathogenic bacteria, the bad bacteria, the types that make you sick. What you may or may not know is that there's a lot of bacteria that don't make you sick. And in fact, a lot of bacteria that you would be more sick living without. Those are the bacteria that make up a huge part of your body. In fact, your body is often more bacteria than it is human cells. And some of those bacteria do things like break down the food you eat. You wouldn't be able to break it down without those. Some of the complex carbohydrates, for example. Some of them release certain types of chemicals that are necessary for the body to function properly. So not all bacteria are bad. The microbiome is just what we're calling the group of all of those bacteria that are present, not in the world, but in either your body as a whole or in a particular region of your body that we're looking at. And the idea of the microbiome and immunologic events meaning rejection or graft versus host disease, that's when the transplanted organ tries to reject your body, which doesn't happen a lot with kidney transplants, but does happen with other types, is that the interplay between the microbiome and your own immune system can change the way that your immune system responds to things in general, because it can lead to either an increase in certain types of T cells, and I'll talk about that in a minute, and it can also change the way that the tone of your immune system is, whether it's highly geared and inflammatory and ready to attack, or whether it's kind of settled down and not in an attack mode, kind of more in a regulatory mode. So for intestine transplant specifically, one of the things that we're looking at, and we don't know the answer yet, is if some component of rejection is due to the change from the immune system that lives in your intestine. In intestinal transplant, the recipient's intestine gets taken out, a change from that to the microbes that live in the donor's intestine. And you know, when the donor's intestine gets transplanted, it comes with all of those bacteria that are living inside of it. And the cells that are living in the donor's intestine wall, the T cells, which are the ones that are responsible for rejection, a lot of them are living in the intestine wall because they're drawn there because the bacteria that they are specific for, and every T cell is specific for some type of antigen, they are specific for the bacteria that live there. And that as the donor's bacteria are lost with time after transplant, then those T cells from the donor that are specific for those bacteria are lost with time after transplant. And about 10% of those bacteria aren't just reactive to those bacteria, but are cross-reactive against other people. So as those are lost, a certain number of those cells from the donor that are also responding to your own body go away. Now, why is that important that we lose some of those donor cells that respond to your body? The reason is because rejection and no rejection, and on the flip side, graft versus host disease, again, that's not your body trying to reject the organ, but your organ trying to reject the body, that's a seesaw. And as one goes up, the other goes down. Rejection is when there's too much of the T cells trying to reject from your body trying to reject the organ. Graft versus host disease is too many T cells from the organ trying to reject your body. A good homeostasis balance is right in the middle. So when you lose too many of those donor cells, then the balance tips. And we hypothesize at least that you will have more of your cells from your body trying to reject the organ than the cells from the organ trying to reject your body. That balance is lost, and that's something that could be responsible for rejection. Now, that's a model that's really specific to intestine transplant, which is not most of you. So let's make this more general. More general, and this is based on things we know, including recent research, especially in the liver transplant world, is that we know that for both rejection and graft versus host disease, which we see often after bone marrow transplantation, that those are associated with the following changes in the microbes in the intestine. So one is either a loss of the good microbes, the ones that help your body, the ones that secrete things that your intestine likes, like short chain fatty acids, the ones that crowd out the bad bacteria. So that's number one. Number two, it could be a gain in the bad bacteria, the ones that crowd out the good bacteria, the ones that directly attack and invade your intestine, the ones that release toxic substances, or the ones that are resistant to many types of antibiotics. That's number two. Number three is just the overall decrease in the diversity of bacteria. So the decrease in the number of species, as that gets lower and lower, that itself is associated with inflammation. And that inflammation that is seen in the intestine, believe it or not, even in non-intestine transplants, is associated with both infection and bleeding and sort of overall poor outcomes, but also rejection and graft versus host disease after transplantation. Why? It's because it's an inflammatory state. And when there's an inflammatory state, it puts the T cells on alert, because T cells don't just see their antigen, which is the thing that they react to, and attack it. They need to see it and also get a signal that says, get activated, go attack. And when that signal pervades in the body, those T cells are just primed to attack. So you see more of all of those immune events, more rejection, more graft versus host disease, more infection. And so that inflammation is really the trigger. And what triggers the inflammation? It's those changes in the microbiome. Again, either the loss of the good bacteria, the gain in the bad bacteria, or the overall decrease in the diversity of the bacteria. And that's what we're looking at specifically with intestine transplant in our lab. And it's what a lot of people have already published with bone marrow transplant, with liver transplant, and now being looked at for kidney transplant as well. These things seem to matter. And in terms of what are the consequences in this for the future, for intestine transplant, they're a lot more specific because there's lots of things we can do to modulate the microbes in the intestine. But what could it mean? And there's also ways that it could be a biomarker for rejection in the future. But what does it mean for kidney transplant recipients, or pancreas transplant recipients, or liver transplant recipients? What it means is that when there's those sorts of events, one potential really targeted therapy in the future is not just throwing immunosuppression at you, but also in trying to reset the microbiome. And that's not just something that is science fiction, that's something we can do. You may have heard already about fecal microbiota transplants. We do it for C. diff infections sometimes. That's just a bad bacteria that can overgrow in the colon and make you extremely sick. It actually is being done sometimes experimentally in the case of graft versus host disease after bone marrow transplant. And theoretically, it's something that could be done in solid organ transplant, kidney, liver, pancreas, for patients undergoing rejection if we find that there's certain changes in the microbiome that are really associated with those events. Not only can we replace or reset the microbiome, we can tailor it even more specifically to the micro changes that we see associated with that. For example, if it's a loss of the short chain fatty acids that feed the intestinal lining from the good bacteria, we can give enemas with those short chain fatty acids. If it's a loss of some of the defensins, which are antimicrobial peptides secreted by the intestinal wall when there's homeostasis between the wall and the microbes, we can give those defensins into the lumen. If we find that it's certain bad bacteria or certain good bacteria, we can specifically go after those bad bacteria or give specifically those good bacteria. So there's lots of targeted ways in the future that these could be novel therapies. We're not there yet with most of the solid organ transplants, but this is what we're talking about today is things that we're not quite there yet. These are the cutting edge research things that will be the future of therapies of tomorrow. So as of now, that's where we are.
So just a question, because this sounds absolutely amazing, and I think we're getting a lot of comments also from our audience. But as you said that we can correct it or we can treat rejection, but are we treating it a little bit blindly if we don't have a sample of the microbiota prior to the transplant versus after the transplant? Since I think there's studies showing that, for example, people who eat, healthy, they have a different microbiome setup versus those who may eat of a more fatty or high carb diet. So, until we start individualizing the microbiome diagnosis, can we really treat people with these kind of therapies?
That's a great question, and this is part of why it's the therapy of tomorrow and not today, because we don't have the answer to that yet.
Yeah.
I would not treat it untargeted, but the goal of what we're doing now is to identify specifically those targets. And once we have those targets identified, then we can do that. And if the particular target happens to be the pre-transplant microbiome, and by the way, we are saving those samples now from intestinal recipients so that if it turns out that's the answer, we can give it, then that's what we give. If it's the defensins, then that's what we give. If it's the short chain fatty acids, that's what we give. If it's a particular species of bacteria, that's what we give. But we need to know what the target is. And then, as you mentioned, there actually are some dietary things that play into this too. And this may be the answer, it turns out. And there's a lot of research that's been happening recently in terms of diabetes and obesity with the diet, but not because of eating too much or too little, but because of the interactions with the intestinal immune system. One of those things was published by Ruslan Menzatov from Yale in the last year or two. And for example, looking at the way certain certain nutrients are seen by a type of a T cell in the intestine. It's a very strange kind of a T cell called a gamma delta T cell. And I'm not going to go too much into this because it's not a little bit off topic, but the way that those nutrients are seen changes some of the way that the cytokines, which are those immune signals from that particular type of T cells, are released into the local area in the intestine in a way that can shape the immune response. So in a way, by eating certain things, it's not a microbe, it's not a antigen, meaning a bad thing in your body that your T cells want to attack. It's just a nutrient that is sensed by the T cells specifically in the intestine that still shapes the immune response.
What is Josh, very nice. What is the likelihood that what you find in terms of rejection of the intestine, where you can do direct studies, and you are doing those, whether this will also translate to the rejection of another organ, not the intestine? In other words, why should it work for the heart or for the kidney or for the liver or whatever else? The liver is sort of close to the gut, but the other organs are not that close to the gut. Do you think that the alteration with cytokine will change the T cell milieu, and that will just recirculate and go everywhere?
There's actually more evidence for that right now than there is for that scenario in intestine transplant, simply because intestine transplant isn't as well studied as the other types. But there is more evidence for that for both kidney and liver transplant right now. So in fact, that's driving the field in intestine transplant, even though it's a little bit counterintuitive because you'd expect it to be more operative in the intestine transplant setting.
And, post-transplant, when the patient is put on heavy immune suppression, I mean, immediately with the ATG and then followed with, various combinations of the immune suppressants, wouldn't that also affect the microbiome and drive it even further beyond the transplant?
The immunosuppression, you're saying? It would, and not just the immunosuppression, but all of the antibiotics that a lot of recipients tend to be on.
The whole package that is being given to a patient in the first year.
Yes, nothing I can say about that other than you've identified a really important confounder in this that makes it really tricky to analyze. That's one of the reasons we don't have an answer yet.
Yeah.
It's a wonderful summary.
Yeah, really, really good. We have a question, practical question from the audience asking, should donors coordinate their diets with recipients?
For intestine transplant, which is largely deceased donor transplant, really for any deceased donor transplant, as you can imagine, that's difficult. For living donor transplant, that would be very interesting. I don't think we have enough evidence to say that yet. But I also, especially for things like anything other than liver transplant, which is really connected to the gut and absorbs a lot of the blood flow from the gut, and so might have some of the microbes. Kidney, heart, lung, by the way, has its own set of microbiome issues because lung and intestine are the two that are connected to the outside world. Right? The lung has the air from the outside environment constantly within it and has tons of microbes from the environment that it's constantly in balance with. That's a big route of entry for those microbes and where they're kept in homeostasis. Same thing with the intestine and the lumen of the intestine. But for the other solid organs, kidney, heart, you're really not transferring a lot of microbes from the donor to the recipient. And I would find it surprising if it mattered that much, but we simply don't know yet till we know better what the targets are.
Thank you. This has been highly interesting, and it's so hard to stop and to actually close this seminar. We would like to continue, but we, out of respect of your time and everyone else, but I think the audience would like to continue. We do need to, I think, consider, Dr. Hardy, to create a whole seminar series with each of these topics as its own seminar, so that we can dig deeper into each area. We do have some call for action here, on the islet side, and maybe a research subject for the intestinal work. And on, of course, the Xeno is really to follow the companies and the academic institutions that are driving the work there. So thank you so much. I'm going to leave it to Dr. Hardy to conclude. And I also wanted to thank Veloxis for the sponsorship.
So I just want to compliment the speakers. I know Adam is flying, so he doesn't hear that, but certainly we were very privileged to hear your views and your presentations. I think that you made them very understandable and very clear. The future is there, not here yet. And I think that there is a promise of better and greater things but that the work has to be really encouraged, and certainly in the case of islets, the public will should really push our regulatory agencies in the right direction. While with xenotransplantation, that also applies, because FDA in many ways is blocking progress, even under really unreasonable circumstances, as viewed by most of us. I was involved in the discussions as long ago as '95, when they were already raising a stink about parvovirus, which never really caused any problems in humans. So, and Josh, I think that this is a whole new field that I think you opened up for all the scientists as well as the patients, and that may be the most promising area of future development in terms of getting rid of, at least partially getting rid of immunosuppression. So I want to thank you for your comments and for your presentations. And I thank the audience for staying with us, and you're still welcome to send questions in. We will try to respond on LifeBulb and Transplant Life to your questions as well as we can, and direct them to the speakers if we can. I just also want to thank Veloxis for their support, and invite you all to the next session, which will be October 18th, I think.
Yes.
Where we will include discussion of transplantation as well as biotechnology and artificial organs, and with experts in those fields. Again, it will be a lesson in medical fantasy, which may reach reality during our lifetime, I hope. So thank you again, and I will say goodbye to all.
Thank you very much.
And thank you to my co-moderator.
Thank you, Dr. Hardy.
Thank you, Dr. Hardy.